So today I am going to continue the last session regarding metatranscriptomics. Here I am going to directly introduce the basic principle and idea of de novo genome sequencing, which I have explained in the previous videos. And there I have summarized an example of metatranscriptomics data processing and analysis. Now this is a summarized presentation for de novo genome sequencing, which is the part of next generation sequencing. So here I will be presenting the example of two reference papers. One is from genome biology and another is from nature. For example, this is one of the paper which is basically presenting the de novo transcriptome assemblies from RNA-seq data, which I have presented in my last video. And this one will be helping those people who are working on de novo sequencing projects, especially RNA and whole genome sequencing. So let's see if you could go to visit this channel Bioinformatics for All and subscribe it. Then you can get the updated version from time to time. Now, what is de novo genome sequencing? De novo genome sequencing generally refers to the process of sequencing a genome without the knowledge of reference genome. The term de novo means from the beginning in Latin. This method is crucial for studying organisms whose genome has not been sequenced earlier. Key features: it builds the genome from scratch, uses short and or long sequencing rates. It heavily relies on assembly algorithms to reconstruct the genome of the particular organism for which the whole genome is not known. Now, in general, the objectives for de novo sequencing are to discover novel genes and regulatory and elements, to understand evolutionary principles and relationships, identify species-specific traits, add in crop improvement, disease research, and microbial discovery. Technologies used are Illumina for short read sequencing, for long read sequencing, PacBio, and Oxford Nanopore. Illumina these days are used for long read sequencing also. Then it is hybrid approaches, which combines both for the better accuracy. Now, in general, the workflow, which is kind of repetitive for the last video. So it includes DNA extraction, library preparation, sequencing, data processing, genome assembly, overlap layouts, consensus or de-bridge graph. Then scaffolding and gap filling, and finally genome annotation. In general, the applications are novel species discovery, cancer genome reconstruction, evolutionary biology studies, personalized medicine, conservation genomics. Now, steps in alignment or mapping includes: get your sequences data, check quality of the sequence data, choose the alignment or mapping algorithm or program, run the alignment, view the alignment, then downstream processing. Now, alignment versus de novo assembly could be visualized as a graphical point of view. Here, for example, short reads could be assembled by using some alignment algorithms, and this could be detail seen in the reference genome database that is available on NCBI database. So there are some other way around also. Sometimes the people, instead of going for de novo assembly, they go for the closely related organism to use their known reference genome to process it here. But it is also used for cross validation of the de novo assembly. So you can also go for de novo assembly and alignment to the reference genome from the closely related organism, and then go for comparison of your de novo assembly. Now, sort read alignments could look like this. As you can see here in the figure, the dashed line is representing this sequence, which is represented here. And then the coordinate of this sort read is mentioned here as chromosome 6. And then this is the starting point. Now, if you want to understand the paper, one of the paper which I have discussed here, evaluation of de novo transcriptome assemblies from the RNA-seq data. Here the authors, Boli et al., has mentioned in the abstract, de novo RNA-seq assembly facilitates the study of transcriptomes for a species without sequenced genome. But it is challenging to select the most accurate assembly in this context. To address this challenge, they have developed a model-based score that is called RSM-eval for evaluating assemblies from the ground truth is unknown. They have shown that this correctly reflects assembly accuracy as measured by ref-eval, a refined set of ground truth-based scores that they also have developed. Guided RSM-eval, they assembled the transcriptome of regeneration of axolotl lib. This assembly compares favorably to the previous assembly method. A software package implementing this method that is called Ketonate is freely available on this link, which you could look over it. And please, before you follow the link, read this paper properly. Now, they have different figures also. What I wanted to show you, these two figures from that paper, that is figure 1 and figure 2. Figure 1 shows the inside way of working of this model, and figure 2 is showing the example to construct true assemblies with different minimum overlap lengths. Then there are some, another figure number 9. This is basically some kind of quality check. How you can understand the true positive, true negative, or let's say truth long only, short only reads by using this method and some other methods. So there is another paper which is from one of the well-known group that is called Mark Vidal, who is really famous in this field, and he has presented one of the main application of de novo assembly, and he has tried to show some of the novel finding by using de novo assembly. That is, they call it as de novo gene prediction. That is here mentioned at the title is saying proto-genes and de novo gene birth. So if you are smart enough, then you could also focus on to discover new genes by using this NGS-based de novo assembly. Then there are some other figures, how to evaluate, analyze, what are the steps involved in order to predict new genes. You could follow up and try to understand these figures, which is presented here. Then there is another figure that is called translation and adaptive potential of recently emerged ORFLs. Now, if I'm again outlining the steps involved in de novo assembly, this which I have described in the previous videos, like workflow for analyzing RNA-seq data. For example, here it is de novo. Then in case of de novo, you need to run fast QC, then go for implement Trinity software to generate a de novo RNA-seq assembly, and then transcript expression, or you can also go for this plus homology prediction. And after that, you need to construct the expression matrix, and finally go for the deck prediction by using different softwares and the tools. So thus you are going to complete. Now, this is figure number 4. which is like from the nature letters. This is like another way of understanding the things. Finally, in summary, you can say the detection of translation signatures, which the Nature paper has described, it is in very short and summarized way. If you are quick to understand, you could easily understand. And first paper, which I have discussed, this paper, there is not big difference. First paper is method development. Second paper is based for discovery purpose. So that is overall what you need to understand. Thank you very much.